So my name is Dima, and I'm a fourth-year grad student in biology. And today we're going to talk to you about genetics, and specifically we'll start with an introduction of genetics. We'll talk about genetic variation, how humans are different at the genetic level. We'll talk about how this relates to disease, and then we'll talk about some new therapeutics that people have been able to come up with based on our knowledge of genetics. So as you know, people are all different, and these differences are caused by a combination of genetics, or what we're born with, and the environment. And so, for example, eye color is something that we're all born with. It's genetic. But then there's other things like our hairstyle, where it's caused by, for example, where we go to get a cut. So that's more the environment. And so the same applies in disease. We have some things that we're born with and some things that are caused by the environment, and the combination of the two. And so before we talk about genetics and disease, we have to introduce DNA. And in order to do that, let's take a step back and talk about our organization. So humans are organisms that are composed of organs, such as the heart or the lungs. And each organ is made up of different tissues, and each tissue is a collection of cells. So the cell is the kind of fundamental unit of life. And each cell has the same DNA. So the DNA is a long molecule that stores information. It's composed of 4 different letters, A, C, G, and T, in different orders. And there's 3 billion letters in a human's genome, or the full collection of DNA. And so each cell has this long DNA molecule that's depicted here. And this DNA has the information for making all the different proteins in the cell. And specifically, one gene is just a stretch of DNA that encodes for one protein. Now, what are proteins? Proteins are the molecular machines in the cell that have different functions. They have these different structures, these different folds, which contribute to their different functions. So a protein might be responsible in transporting a molecule from one place to another, or in metabolism, they degrade certain molecules from large molecules to small molecules. And so there's all these different proteins, and the instructions for making each protein are encoded in the DNA. And so this is what's referred to as the central dogma of molecular biology, where you have the DNA, which has the instructions. Then there's an intermediate called the RNA, which is really just a copy of a portion of the DNA. And then that encodes for a protein, which has a 3-dimensional shape. And so by analogy, if you think of an instruction book that has the instructions for making all of the different parts of the car, that's like the DNA. And then the RNA is a copy of one page, so maybe a photocopy of the instructions for making a wheel. And then the wheel is a protein that has a protein that has a specific function. And so a little over 10 years ago, many of you have probably heard of this project called the Human Genome Project, where we're actually able to get the full sequence of DNA, 3 billion letters for one human. And now we're getting genomes for all different people, which have small differences that Mike will tell you about. And so before we move on to that, how humans are different, and start talking about disease, I'll take any questions about the basics of genetics and molecular biology. Yes?
Looking at the list of terms, is there a term for the between gene sequence that like CRISPR would use?
Yeah, so CRISPR is in bacteria, so we won't be touching on that. Yes, so the DNA in humans, there's genes, but there's also regions that are called intergenic regions, which don't encode for proteins. So
it would be intergenic that bacteria use?
So for CRISPR specifically, yes, it's used in that. Yep.
Are some of the intergenic regions like just stuff that they haven't figured out what their genes for yet?
So we think we know all the genes that code for proteins, but there's regions that we don't know their function, and some of them code for RNA that doesn't make proteins, called non-coding RNAs, and then other regions don't code for any RNA, but might have other functions. Yep?
Can you explain the difference between introns and exons?
Sure. Sure, so it's a bit beyond what we'll talk about today, but a messenger RNA has introns and exons, and so these are different portions of the mRNA that are joined together to change some of the things about the protein. So in reality, one gene can make more than one protein or slight variations, but that's, yeah, we won't go into that today. All right.
Maybe I'll tangle that. Okay, great. Is this already on? Can you guys all hear me? All right. Hi, I'm Michael. I'm a grad student, a third-year grad student in the Biological and Biomedical Sciences program. Today I'm going to tell you about genetic variation, genetic basis of disease. I'm going to build on what Dima has told you about how genes work and how proteins work, and talk about how we're different at the genetic level, how this can lead to disease. And so we'll first talk about what a genetic mutation is. You may have all heard about mutations. We'll start off with a mother and a father. I'm going to color the mother's genes. This is a gene, a segment of DNA that has a certain base pair sequence. And color hers in pink and the father's in blue. And you'll notice that the sequence is largely the same between the mother and the father. Also important to notice that each person has 2 copies of each gene. One they inherit from their mother and one they inherit from their father. So they're largely the same, except for the mother sequence has 1 base pair that's different. Most of their genetic sequence is completely the same, but this mother has only 1 copy that's different. And so this mother and father will have children, and they'll pass on some of their genes to each child. So each child will get 1 copy from the mother and 1 copy from the father. So this daughter here got 1 pink copy from the mother. She happened to get the copy that has a T here, and she also got a blue copy from the father. Since the father's copies are both the same, it doesn't matter which copy she got. The son here got the mutation, the A instead of the T, from the mother. It got the normal blue copy, and the fathers were both the same. So this is how genes get passed on from one child, from parents to children, and so forth. And so there's 2 terms that we're going to throw around a lot today and throughout the rest of the talks, and that's genetic variants and genetic mutations. And scientists all kind of disagree on exactly what these terms mean. And so today we're going to use them completely interchangeably. But genetic variants is any mutation, any DNA change that's present in the population. And the mutation, we're going to call it genetic variant with effect. So genetic mutation is a subset of genetic variants. And we'll use them completely interchangeably today. And to go back to the central dogma and talk about how genes can lead to disease, so this is what Dima talked about, the DNA leading to RNA, which is the kind of a photocopy of 1 page of the DNA, which is a book that can code for a protein. And so in disease, let's say you had a mutation in a gene. I'm going to use the example of sickle cell anemia, a really rare blood disorder. And here you have, by the yellow star, you have a mutation in the genome. And with a photocopy, that mutation becomes propagated to the mRNA level. And so the protein, sometimes this mutation in the genome can lead to a misfolded protein. In this case, you see the yellow circle instead of the purple. And so this protein becomes misfolded in the case of sickle cell anemia. What happens is that normally these proteins, red blood cells are spherical, and when these proteins misfold within the red blood cell, it causes the red blood cells to turn into this really weird sickle shape. And these red blood cells are less capable of carrying oxygen, and they actually get blocked up in little blood vessels. And these patients end up dying at a really young age. So going back to the slide that Dima showed, so humans are largely identical. But if you look around the room, there's differences between you and I. There's different skin colors, difference in hair color, hairstyles, and so forth. And at the genetic level, we're 99.9% identical to each other. That means that between you or I, or any 2 people in this room, we're different on average 1 in every 1,000 base pairs. Now, the human genome is 3 billion base pairs in length, and that means on average, each person carries about 3 million mutations in their genome. And that's a lot of mutations. Unfortunately, I'll tell you in a couple slides that most mutations have no effect. So if we had 3 million bad mutations, we'd all be really screwed. And this is true across populations. So the genetic variation is shared within, genetic variation is largely shared across populations. So we're much more similar than we are different. And that's really important to remember as you think about genetic differences between populations. And so this is how, and so genetic variants can also be common or rare. So this is the reference sequence for a gene. This is the sequence that the Human Genome Project has determined to be what we call the reference, what we compare everything to. And as Dima mentioned, we could sequence people. Here we sequence 10 different people. And you can see at the first position, everyone has the exact same A as a reference. However, as we move along, we see at this fourth position, the reference shows a T, but only half the people have a T and half the people have a difference, or an A. So this is what we call a common genetic variant. It's present in 50% of the population, have a difference from the reference. And you can move along to this other base pair in the same gene, and you can see that the reference shows a G, but 9 out of 10 people in the population also have a G, but one person has a difference, which is an A. So this is what we call a rare genetic variant. It's present in only one person. And so genetic variants can have a whole range of how common they are. They could range from being present in one person in the entire population of 7 billion people, or they're present in half the people in the population. And genetic variants can have a wide range of effects. So if I say the green car here is normal, we could have the vast majority of genetic variants in the population, 99.9% of them are also completely benign or have really tiny effects. So you can imagine a genetic variant that changes the color of the car from green to blue, has absolutely no consequence for the function of the car. And some genetic mutations are actually beneficial for you. They can make this car go a little bit faster or something like that. And there's also some genetic mutations that are damaging. This often occurs in the context of disease, which we'll talk about in the next section. So just to summarize what Dima talked about, this is kind of the foundation for today's talk and for all the talks today, are the genes code for proteins. And genes are these segments of DNA that have these 4 base pair, these DNA sequences that code for proteins. These proteins perform the cellular functions. And individuals differ at some sites of DNA, about 1 in every 1,000 base pairs, we differ from each other. And that these genetic variants can be common or rare. They can be seen in one person in the population, or they can be seen in many people. And most of these genetic variants, the vast majority, are completely benign or have very small effects, but some can be beneficial or damaging. Yeah, I'll take any questions. Yeah.
When there's a variant that affects a protein, changing and folding the main way that the proteins get affected, as opposed to like a different molecule?
Sure. So the question was, with a DNA change or mutation that affects a protein, how does it really affect the protein's function? So there's actually many ways it can affect the protein's function. One way is for it to misfold. Other ways it could have a new function. This is what we call, these are gain-of-function mutations, which we'll talk about in the context of cancer. It could also cause the protein to be completely absent. These are what we call truncating mutations. They can also change the expression level of protein. So there's many different ways, and we're going to kind of talk about the simplest example, which is a misfolded protein. Any other questions before we move on to the next section? All right. So now we're going to talk about how we move from genes to disease, how these DNA base pair changes can cause disease. And so I'm going to classify disease into 3 broad categories. This is a really broad categorization. So first, we would call purely environmental diseases. These are examples like snake bites or car accidents, where your genes have little or absolutely no consequence of whether you're going to get a snake bite or go in a car accident. Second class diseases are purely genetic diseases. These are usually rare diseases with a single gene that causes disease. An example is sickle cell anemia. You can see these red blood cells here that have a really weird sickle shape. And these are really rare debilitating diseases. An example may be cystic fibrosis, which you might have heard about. And there's some diseases where the genetics and the environment are both important. Examples might be cholesterol levels or obesity, where your genes and your diet, exercise, both impact whether or not you'll get the disease. And today I'm going to talk about these latter 2 categories that have a genetic basis. So to classify those 2 genetics, so the first category is what we call rare monogenic diseases. These are purely genetic diseases. The other category we call common polygenic diseases, which are ones where genes and environment are important. I'll go into more detail about each one of those. So in the rare monogenic, the name suggests that monogenic means single gene. So it's usually a single mutation of really large effect. If you have the mutation, you're going to have disease. If you don't have the mutation, you won't have the disease. And here the environment's less important. So no matter what diet you eat or how much you exercise, if you have the mutation for sickle cell anemia, you're going to get sickle cell anemia. Your environment's much less important. So examples will be, as I mentioned, cystic fibrosis or sickle cell anemia, Duchenne's muscular dystrophy. These are all really rare, debilitating diseases with really early onset morbidity for most people with the disease. And then there's common polygenic diseases. So these are usually more common diseases, and polygenic just means poly means lots. So lots of genes are involved. And here, there's many genetic variants, many genes, many mutations in many genes, oftentimes thousands of genes that are involved in the disease. And each of these genetic mutations confers a tiny bit of risk to that disease. But added together, they can have a substantive increase in risk. And here, the environment's often very important for whether or not you get the disease or not. And examples might include type 2 diabetes or obesity, where both your genetic makeup and your environmental contribution to diet and exercise are important. To kind of illustrate all these examples, Dima and I use the example of LDL cholesterol, which you might have heard about from your doctor. So I'm going to go into a little brief introduction about how cholesterol works. The details aren't too important, but just to make sure everyone's acquainted. So cholesterol is present in foods. It's particularly high in fatty foods such as pizza and hamburgers that we all love. And so cholesterol is a fat, and fat is not soluble in blood, so that's to be carried in different ways. So it's carried in what's called a lipoprotein, which is a protein molecule bound to many of these lipids or cholesterol molecules. And one example of a lipoprotein molecule is a low-density lipoprotein, or LDL. You might have heard about this as being the bad cholesterol. And so all LDL is just basically, it's a bunch of proteins bound to a bunch of cholesterol molecules that allows the cholesterol to be carried in the bloodstream from one compartment to another. And eventually goes to the liver. And the liver is really the center where most of your cholesterol is metabolized. So cholesterol is bound in the blood as an LDL molecule, and it gets to the liver cell. And on the surface of the liver cell are these LDL receptors. And LDL receptors are proteins on the surface that can bind and uptake the LDL molecule. So they have a shape that allows it to bind specifically to the LDL molecule and uptake it into the liver cell where it's metabolized. And you might have heard again that LDL is bad cholesterol, and that increased LDL leads to heart attacks. And so what happens is LDL can actually build up in your blood vessels. And this is kind of a view of the blood vessel. And if you have enough LDL, it could build up as a plaque right here shown in yellow, and it could eventually block off most of that artery or that blood vessel and lead to decreased blood flow. And if this happens in one of the blood vessels that supplies the heart, this could lead to a heart attack. And so we've probably heard that increased LDL levels are associated with highly increased heart attack risk. They actually cause increased heart attack risk. If you're able to lower your LDL cholesterol, you can decrease your heart attack risk. And so if you look at the population, there's a huge amount of variation in the LDL levels. And so here on the horizontal axis, I have the LDL levels. The numbers aren't too important. On the vertical axis is the percentage of people with that level of LDL. So here, each bar just represents the percentage of people with that level of LDL. So you can see that for LDLs between 80 and 90, approximately 10% of people will have that level of LDL cholesterol. You might notice that this is a bell-shaped distribution. This is actually a really nice bell-shaped distribution, or normal distribution. And that means that most people have LDL cholesterols near the middle, and only a few people have LDL cholesterols in the extremes, very high or very low. And this distribution is actually the result of both the genetics and environment. It's a contribution of genetics and environment to the variation in the population LDL cholesterol. So in the case of LDL cholesterol, about half the variation is contributed by genetics and half is contributed by environment. So if we had the case that everyone had the exact same environment, so everyone had the exact same diet, lived in the exact same place, exercised exactly the same, and the genetics were the only thing that were important, the genetics only contribute to half the variability. So this curve would be about half as wide. So many more people would be near the center, and the width of this curve would be cut in half because half the variation in the population is contributed by the genetics and half by the environment. And so why do we do genetics? So in the past few decades, we all know that LDL cholesterol leads to heart attacks. So we want to lower LDL and try to reduce heart attack risk for people. And so we have to understand how LDL cholesterol works first. And through decades of research, we've done many elegant studies in many cell lines and mice and fruit flies, model organisms, to understand how these work. And we found a few dozen genes. So a lot of really hard work went into this. However, we really want to treat LDL cholesterol, to decrease heart attack risk, we want to understand LDL cholesterol in the humans at the body level. And so we want to find genes that are relevant for LDL cholesterol in human beings. And so a lot of these things that you find in fruit flies or model organisms may or may not be relevant for humans. And this is really important because by finding the genes, you can understand disease and you can find new drug targets. And so how you do genetic studies, you're going to find mutations in genes that alter the cholesterol levels in humans. There's two types of genetic studies that we'll talk about today. One's called a genome-wide association study, and one's called a linkage study, and I'll talk about each one of those in detail. So genome-wide association, or GWAS, it's a large study that you perform in many numbers of people. You do this in general population to understand what genes contribute to the variation in the general population of LDL cholesterol. And because you have so many environmental contributions, each gene only contributes a small amount of risk. You need many people to find these genes. And I'll tell you that there's actually the size of the population that you need to find some of these genes in a couple slides. And so you look across the genome and find genetic variants that are associated with differences in cholesterol level. And so for each common genetic variant, so these variants present in many people, you can test whether the variant is associated with cholesterol or not. I'll show you this in the next 2 slides. So here's a reference sequence for this gene. I'm going to sequence 10 people. And I'm looking, again, people are largely the same, except at this 4th position. Again, here half the people have a T and half the people have an A. So again, this is a common genetic variant. Half the people have the T, half the people have non-reference. And so what I'm going to do in a genome-wide association studies, I'm going to test what is the distribution of people who have an A, what is the distribution of people who have a T, and I find that people who have the T at this position have on average a cholesterol level of 100, which is the average in population. People who have the A at this position have on average 120. And so having an A here is associated with a 20% increase in levels of cholesterol. So we call this genetic variant to be associated with cholesterol because it's associated with 20% increased level of cholesterol. What's really important to know is that these curves, so the red is the curve of people who have an A, the black is the people of the curve who have a T, that these curves are overlapping. So that means that this single genetic variant does not explain your cholesterol levels. You could have a T, which is associated with lower cholesterol, and still have very high cholesterol out here. You could have an A, which is associated with higher cholesterol, you can still have very low cholesterol. So this genetic variant only explains a small amount of why your cholesterol is high or low. And we go to the 2nd position in the genome. This is a different gene. Again, I find here that half the people have the reference, which is a G, and half the people have non-reference, which is an A. Again, I'm going to ask the same question, what is the average cholesterol levels of people who have a G versus the average cholesterol levels of people who have an A, and I find they're actually identical. So this genetic variant is not associated. So my goal in genome-wide association studies is to find the variant ones that were associated with cholesterol and weed out the ones that weren't. So what have we found from GWAS? And I told you that I do it in a large number of people because it's such a messy experiment. There's so many other factors going into it. So the latest genome-wide association study was done in almost 200,000 people, 200,000 patients. It's a huge, involved many hundreds of scientists across the country, across the world. It's a huge collaborative effort to make this happen. And over the last couple of decades, we found a couple dozen genes involved in cholesterol, but this one study found 157 genes. So it's probably quadrupling the number of genes that are now known to play a role in cholesterol metabolism. And just what we call a positive control, to make sure that our experiments were done correctly, that we're finding the right things, we do find many genes that were previously known to play a role in cholesterol. So we know we're on the right track. We also know some of the genes that we found are also the drug targets. So we know we're on the right track. We're not finding things that are just completely off. We also find many, many new genes, hundreds of new genes, about 100 new genes that have never been never thought to have a role in cholesterol levels. This does represent new biology, new biological mechanisms. They also represent new drug targets, which Dima will talk about. Now the second flavor of genetic study is what we do a linkage study. We do this in the context of really rare genetic diseases where the gene explains all the risk. And I'm going to talk about a genetic disease of cholesterol metabolism called familial hypercholesterolemia. It's a long word, but it's not that daunting. Familial means it runs in families. Hyper means increased, and cholesterolemia means cholesterol in the blood. So just people with really high cholesterol levels in their blood that run in families. And it's a rare disease. It's not that rare. It's about 1 in 500 people have this disease. And these people have very high cholesterol levels. Their cholesterol levels actually are off the distribution. It's so high, it's not even on the curve anymore. And it's so high they actually can get deposited in their skin, on their eyelids. And what this is really important is that the individuals, because their LDL cholesterols are so high, they get heart disease and they get heart attacks by the age of 60. So it's a really bad disease. And how we're going to do this, we're going to find the genes for this kind of disease in a linkage study. We're going to recruit families where we find many individuals with this disease. And this is what's called a pedigree. So in the circles are females, our boxes are males. So this is a mating between the mother and the father, and they have 5 children. And if the box is shaded, that means that the individual has familial hypercholesterolemia. And if the box is not shaded, that person is normal. And so we're going to find which mutations are linked to diseases. Again, this is a linkage study. So we're going to find out which mutations are present in only the people who have the disease and not in the people who don't. And because the genetics, a single mutation of large effects that cause you to have disease, we're going to only look for these rare mutations that have a huge effect here. And remarkably, if you do this study, you're going to find genes, you can find mutations in the gene that codes for an LDL receptor, which talked about uptakes LDL from the bloodstream into the liver cell. So here's again the normal LDL receptor. You have these LDL particles that can bind to the receptor and go into the liver cell to be metabolized. But in these individuals, the LDL is still in the bloodstream. At the liver cell, their LDL receptor has changed their shape. So instead of having that ability to bind the LDL cholesterol, they have a new shape that can no longer bind the LDL cholesterol, and it can no longer be uptaken into the liver cell. So it builds up in the blood and forms plaques. These people get early onset heart attacks. To summarize this section, so for rare monogenic diseases, these are genetic diseases where a single mutation causes you to have disease. There's just, the genetics plays a primary role. The environment's much less important. We're going to find these mutations by linkage studies. For common polygenic diseases like type 2 diabetes and cholesterol in the general population, the genetics and environment are both important. And we're going to find these genes by genome-wide association studies. And these genes can be found through these genetic association studies through many, many different ways in large populations and small families. And these genetic studies have unveiled many new drug targets and also helped us understand the biology that regulates LDL cholesterol metabolism in humans. And I'll take any questions for this section. Yes?
How you can identify variations which participated into the for a particular disease, which could be significant causes.
Yeah.
But how do you identify whether that gene has other effects? Maybe some of them either benign or even beneficial for some other things.
Yeah.
How do you do that?
Yes, so the question was we can find genes, mutations, genes that have cause disease, but what if these genes and mutations have other effects? It turns out that most of the genes have many functions in our body. And we do see other effects. And so this is something that we can find through genetics by looking from the genes, finding the mutations first and looking at what the effects are. And so we can definitely find genes that have all these effects. It's actually really important for finding drugs. So each drug targets a certain gene or protein. And so as you know, a lot of the side effects of drugs are due to if you take out a protein that has many functions. And so through genetics, if we can actually find genes, if we find mutations in genes that destroy the function of that gene, we can find out what the effects are ahead of time. So if we took out that gene through a drug, we would know what the side effects are. So that's how genetics be incredibly powerful to answer these types of questions. Yes?
In the large-scale population studies, how can you actually prove that this particular gene is causing the effect? Correlation, not causation. Right,
so the question is how can you prove that? It's true for both the linkage study and the association study. So these genetic variants can be associated with disease, but by not necessarily cause disease. And so there's one really important thing about genetics is that there's the arrow of time, and that you're born with your genetics, and everything else comes afterward. And so we know that the genetics came first and disease came second. That's something really special about genetics. But we don't know how it causes disease. So it could be correlated through disease with some really weird mechanism. So these studies, we try to control for those mechanisms. We control for other factors that we think might be associated. So these genetic variants might be associated with what you eat. It might be associated with whether you like hamburgers or not. That could explain why you have high LDL cholesterol. And those things are difficult to control for. And those things, they come up, and that's why you need to do the follow-up studies to really understand how these genetic mutations lead to disease. That's where the mouse models come in afterward. Yes?
How do you go about identifying less obvious environmental contributors?
Sure. So so the question is, how do you go about identifying less obvious environmental contributors, like whether somebody likes to eat hamburgers or not? It's very difficult to identify it at a large scale, and it's hard to do. And this is why you need large numbers of people to kind of wash out these environmental effects. But we definitely control for kind of the obvious ones: your gender, your age, your ethnicity. And some of the more subtle ones are very difficult to control for, and that's one weakness of a genetic study. That's why we still need to do the follow-up biology. Yes?
Does controlling for it mean like adding little math? Like, I mean, is it pretty much just equations in there?
Yeah, so there's 2 ways. So one is you can try to find, so you basically for disease, let's say if we're trying to find genes for type 2 diabetes, how will we control for that? So we're going to look for, our cases are going to be people with type 2 diabetes, and our control will look for people who are roughly matched by ethnicity and gender and age. That's one way to control for it. And then the other way is through math. We could do what's called covariate, which we won't explain today. That's a way that you can try to take away those other factors. Wait, any other questions? All right, I'll hand it off to Dima again.
All right. So for the last part of our talk, I'll talk about how we can use our knowledge of genetics, which Mike talked about, to actually design new drugs, new therapeutics to treat disease. So many of you have probably seen this movie or heard of it, X-Men, where you have these mutants who have these rare superpowers. So some of them, for example, there's one that can fly, there's one that can shoot lasers out of his eyes, and of course, we don't have any humans that can do those things, but actually it turns out that there are rare humans that we can study, sometimes just 1 or 2 people in the whole world who have these very unusual traits. And so, for example, there's people with a mutation in a gene called CCR5, and those people don't get HIV, so they're resistant to it. They don't get AIDS. And then there's a mutation in a gene called PCSK9. We'll talk a lot more about this one. And these people are quite lucky in that regardless of what they eat, their chance of getting a heart attack is much lower. In some cases, 90% less than people without this mutation. There's another gene called LRP5. When you have a mutation in this gene, you have very, very strong bones. They almost never fracture. And then there's a mutation in a gene called myostatin, where people with this mutation, they are just born with abnormally large muscles. So here, for example, is a bull that has a mutation in myostatin. This person next to him does not have this mutation. It's for comparison. And actually, it's also present in people. So here's a boy who is just born with these abnormally large muscles. And going back to the PCSK9 mutation, so again, there's a small number of people that have a mutation that completely inactivates the PCSK9 gene so that it's not active. And it turns out that these people have much lower, on average, blood cholesterol LDL levels. And again, this greatly reduces their chance of heart attacks. And so biologists, this is quite a recent discovery, and biologists became very interested in why this is and what's the actual molecular mechanism for why having this inactivating mutation has this amazing effect. And so here's one study with a small study where they looked at a specific mutation that completely inactivates the protein, and you can see there's about a 90% reduction in the percentage of heart disease in people with this mutation compared to people who don't have the mutation. All right, so in the past few years, people have actually figured out why this works, why inactivating PCSK9 gives you this effect, and it goes back to cholesterol. So as you remember from Mike's talk, you have these liver cells with LDL receptors that bind LDL with cholesterol from the bloodstream. And so they take up this LDL cholesterol and take it out of the bloodstream. Now it turns out that PCSK9 is this protein that's also present in liver cells. And this protein, its normal job is to eat up some of these LDL receptor proteins. So it's a protein that chops up other proteins, but specific ones. And so this PCSK9 protein will eat some of the LDL receptors, destroying them, but not all of them. And so now when you have less LDL receptors, what this means is that less LDL can bind from the bloodstream and enter the liver cell. So what this means is now there's actually more LDL in the bloodstream, which can clog up arteries. And so we know that PCSK9 affects LDL levels through this mechanism. And so normally, you have, this is the central dogma, where you have DNA makes RNA and protein, in this case PCSK9, and if you have a mutation, then that mutation can be carried to the RNA, which misfolds the protein. And here, the shape of the protein is changed. So here the Pac-Man doesn't have an open mouth anymore and can't degrade the LDL receptor proteins. So this mutation changed its structure such that its function is now changed and it can't eat LDL receptors. So what does this do? What this does is if the mutated PCSK9 cannot eat the LDL receptors, there's more LDL receptors on liver cells. And that means that more LDL can be taken out of the bloodstream into the liver, compared to normal people who do have this gene. And so you have lower cholesterol and LDL levels in the blood if it's mutated. And so pharmaceutical companies recently, after this finding, became very interested in this and thought, what if we could use our knowledge of genetics of these people, these rare people who are naturally lucky in that they have a much reduced probability of heart disease, and we can actually design a drug that mimics that effect. And so what they wanted to do is see if they can, in people who have the normal PCSK9 that can degrade LDL receptors, see if we can block it. And so what two pharmaceutical companies have done, and these drugs have finished pretty much almost all of the clinical trials and will likely be available in the next year or two in the clinic, is they made drugs that block this protein. So what happens is the drug actually physically binds to PCSK9 protein and blocks it such that the protein cannot chew up LDL receptors. And so it turns out this drug is very effective. It, again, mimics this natural mutation, and it turns out in these clinical studies it was shown that it's better than any other cholesterol-lowering drug. So for example, currently there's a class of drugs called statins, such as Lipitor, which is the best-selling drug that lowers cholesterol levels. But for some people, it doesn't work very well, or it has a mild effect. And these PCSK9 inhibitors actually have a much stronger effect than any of the currently available statins. So many cardiologists are very excited about this drug, and our knowledge of how to make it came from genetics. All right, so to conclude our whole talk, we told you that there's differences in DNA between people, and that these differences, together with the environment, contribute to disease. We also mentioned that there's genetic variants that are associated with certain disease risks, and we can find them using genome-wide association studies or linkage studies. And we can use our knowledge of genetics from some of these studies to actually design new drugs to mimic the effects of these rare people that have beneficial mutations. And with that, we'll take any more questions. Thank you. Yes?
So why does PCSK9 exist at all? Why would you need to clean up LDL receptors? And if you have that genetic variation so that you have many LDL receptors, can you have too many? I mean, what's the disadvantage of this if proteins have multiple purposes?
Right, that's a great question.
The downside.
Right, so to repeat the question is why is it bad in any way to have a mutation in PCSK9? Does PCSK9 have other functions, and why don't we all have it so that our cholesterol levels are lower? And that's a great question, and the answer is we actually don't know the full answer to that question. So PCSK9 may have other functions as well that we don't know of, and there's many people now studying it. But one of the things that makes pharmaceutical companies much more confident in developing a drug for this is that these people seem fine. So of course there could be that they have some increased risk of something else that we don't know about. But actually, as far as we can tell so far, and again, this is a fairly new area of study, only about 15 years since finding this, there haven't been any adverse effects recorded in these people or any other increased disease risk. So it seems safe, and we actually don't know if there's any why it was selected against, for example, in evolution. Yeah. But that makes people much more confident in seeing that some people have this mutation to develop a drug, because otherwise, if that's not the case, there's always a worry that the drug will have some side effect.
Because those people may have another as yet undiscovered genetic mutation that prevents PCSK9 from affecting them negatively. We don't know that.
That's true. So there could be a lot more going on. But that's why there's a lot of studies following these people. Other questions? Yep.
When you say PCSK9 to like other biologists, how do you indicate you're talking about the gene versus the protein, or does it matter?
The question is about when we use PCSK9, does it refer to the gene or the protein, right? So a lot of people use that interchangeably, but yes, so both things are PCSK9, referred to as PCSK9, but there's the DNA that encodes the protein, right? So it kind of depends on the context if we're talking about the DNA or the function of the protein. Yep.
In the research today, or the findings today, if you're directing so much LDL cholesterol, is the liver able to process it well if you're inundating it with more than it's used to? Are they finding the liver is being adversely affected by all the LDL cholesterol? Taking it out of blood, but bringing it to the liver, is the liver able to process it?
So sorry, the question is in people in normal people?
Because you inhibited the drug target, so you're directing the LDL cholesterol out of the blood, but are you overwhelming the liver in any way?
That's a great question. Yeah.
Process it.
So the question is whether when you block PCSK9, you have more LDL receptors and then take up the LDL into the liver. Is that bad for the liver? And so there's just been this giant clinical trial of tens of thousands of people that had this inhibitor, and it didn't seem like that was the case. So they looked at a lot of markers related to liver function. It didn't seem like that was the case, but it could be long-term that there are some effects. So that's something people are studying. But from the initial clinical trials, it doesn't seem to be the case. So it seems like the liver can handle it. But yeah, great question. Another question. Yep.
So can I have the name of the pharmaceutical companies that are doing this? Because I need to buy stock.
All right. So actually, actually the stock has gone up based on this. So one company is called Regeneron, and then another company is called Amgen, and they're actually competing against each other. So they're both racing with this drug.
There's actually an FDA meeting next week on the Amgen drug.
Yep.
But I don't know if that's this one or not.
Yep.
And then also Sanofi is supposed to be.
Right. So Sanofi is working with Regeneron.
Okay, I think we're going to have to wrap up and move on to our next talk. But if you have more questions, Dima will be around at lunchtime, so feel free to talk to him or Mike as well. Let's give them a round of applause.